everyone. We're going to talk about Chapter 18, Practical Applications of Immunology. We'll talk about some immunological diagnostic tests. This is a lot like what you did in lab with the immunological tests. A lot of those tests will be discussed here. All right, so for a lot of those lab tests that you did, you needed monoclonal antibodies. So we talked about how B cells, as part of your acquired immunity, produce antibodies, and they only produce one type of antibody. So the antibody that they produce is a lot like their receptor, except it's just lacking that small portion, the transmembrane domain, which anchors that protein in the membrane of the cell. And so those antibodies are secreted from the cell, and it's just one type of antibody, and they can bind to the antigen and neutralize it and initiate complement, et cetera, et cetera. So this is, we're talking about how you would produce a monoclonal antibody in vitro. And you might remember from when we talked about tissue culture that you can get cells directly from the body, and they will go through a couple, 2-3 cell divisions, but then they die after that. So the idea here in producing a cell line, in vitro, that can produce one type of antibody, or a monoclonal antibody, is you would need to make those B cells that are producing the antibody immortal. And so I'll go through the procedure, but first of all, you would need to, immunize a mouse against the antigen, have it start to produce the antibodies, and then harvest those B cells from the mouse, fuse them with a cancer cell, to make this immortal hybrid cell that you could grow in culture indefinitely that would make monoclonal antibodies. Okay, so here's the figure. So the mouse would be injected with an antigen, and we talked about what types of antigens would be best at eliciting an immune response. Those would be like proteins, glycoproteins of a certain size. So the mouse would be injected. It could be an inactivated toxin, a toxoid, and then the mouse would raise antibodies against that particular antigen. So in the spleen, you're going to have a bunch of B cells that are going to be producing antibodies, and they could be, remember when I said that an antigen has different epitopes, and each one of the B cells, the vast array of B cells that you have, has a slightly different receptor that can bind to these different epitopes on an antigen. You will get from this spleen multiple, not that many, but several B cells that produce antibodies against epitopes on for the particular antigen that was injected. Now remember, each one of these B cells is only going to be producing one type of antibody, the monoclonal antibody. And then you want to fuse those spleen cells to the myeloma cells, the cancer cells, so that you can make the immortal hybrid cell line, the hybridomas. So you mix those spleen cells with the myeloma cells, and then as a result, you're going to get some of these hybrid cells. You can put the hybrid cells into selective media. Okay, the spleen cells are eventually going to die because they are only going to, that's the problem with them, right? Is that they're not immortal. The selective media actually selects against the myeloma cells so they don't grow. And the only cells that are going to grow are going to be these hybrid cells, okay, the hybridomas. All right, and then you can, separate them out, okay, because remember they could be making antibodies to, these different cells could be making different antibodies to a particular antigen, to different epitopes on an antigen. So you would culture them separately, and basically these hybridoma cells are immortal B cells, which are going to be making antibody. All right, and the antibody would be secreted into the culture media, and it could just be basically removed from the culture media. And if you just did a couple of purification steps, you would have the monoclonal antibody that you can use for many of the diagnostic tests that we are going to be talking about. So this a monoclonal antibody, and this is a human monoclonal antibody. I just wanted to give you this example. It's used for breast cancer, and it's called Herceptin. Years ago, about 20 years ago, Genentech developed this antibody, and when they used it for people who had breast cancer, there was no like nasty chemotherapy or radiation. The people just basically went in and got hooked up to an IV of antibodies, and sure enough, for the particular type of breast cancer that they had, and these would be like the human EGF receptor, HER positive type breast cancer, Herceptin was able to shrink those tumors. Okay, so definitely can be a good therapeutic as well, these monoclonal and here it is. The precipitin ring test is for soluble antigen. Okay, but here it's the same idea, okay, but this is an agglutination reaction where you have particulate antigen. Okay, so here you've got like this whole huge big cell that is being bound by antibodies and clumped together. Okay, and the same kind of rules would apply, like when you have an excess of antigen or an excess of antibody. At the optimal concentration, you would get agglutination. You would get these clumping of the particulate matter together. Okay, in this case, it would be cells. Okay, remember we said that IgM, because it has 5 antibodies, okay, held together by the J chains, it is going to be the best at precipitating because it's got 10 binding sites, right? Each one of the antibodies is bivalent. Okay, here are some agglutination reactions that are similar to the reactions that you did in the lab exercise, okay, that you looked at for the PowerPoint. Okay, so in the first part of this figure, you would be detecting the antibody, okay, so the latex beads are covered with the antigen, if you have the antibody, if the antibody is present, it would clump those latex beads together, okay, and you would see agglutination. Okay, so you would see the clumping. So another type of agglutination reaction that you could do would be where you have now, instead of the antigen bound to the latex beads, you have antibody bound to the latex beads, and you are trying to detect the antigen. Okay, so in the first part of this figure, you would be trying to detect antibodies from the patient. In the second part of this figure, you would be trying to detect antigen from the patient. Okay, so if the patient has a particular bacterium that these antibodies are specific for, okay, when you add the agent to the beads coated with the antibodies, okay, the beads are going to clump together. It would be a positive agglutination reaction if the antigen is present. So this is looking at agglutination again. Okay, it's hemagglutination, all right, because these are red blood cells in this well. And it's looking at how you could measure an antibody, okay, concentration, okay, or titer. And so what they did in these wells was they added different dilutions of the antibody. Okay, so a 1 to 20 dilution, then a 1 to 40, a 1 to 80. Okay, so these are 1 to 2 serial dilutions, right? Okay, 1 to 2 serial dilutions. And if you look at the bottom panel, okay, where we're looking at the side view of the wells, when the antibodies are bound to the red blood cells and they're agglutinated, the antibodies actually kind of hold the red blood cells out at arm's length, kind of, so that they don't all just kind of precipitate. Well, that's probably not the best word to use here, but they all don't kind of just roll to the bottom of the well. Okay, they don't just because the wells are sloping, right? So if they're not bound by the antibody, okay, they would all just basically, and they're not held apart, they would just basically go to the bottom of the well, and that's what it would look like on the right side of the figure, okay, where they're all just at the bottom of the sloping well. So you can kind of see how, okay, the lowest dilution, 1 to 20, would be the highest concentration of antibody, right? So you can see that you've got like a good concentration of antibody at 1 to 20. It's still good at 1 to 40. It's still good at 1 to 80, 1 to 160. But then when you go to 1 to 320, you don't have enough antibody to agglutinate the red blood cells. And so they all just kind of, they're non-agglutinated. They all just kind of roll to the bottom of the well. Okay, so by doing this, you can actually measure an effective antibody titer. Okay, so that's hemagglutination when you're looking at the clumping together of red blood cells. Okay, so the heme part of that. Viruses can cause hemagglutination as well, And if you have an antibody to a virus, you can prevent hemagglutination. Okay, so that would be like a neutralization antibody, right? Because that antibody would be preventing the virus from binding to the cell and causing its deleterious effects. Okay, so here is hemagglutination caused by a virus. Okay, so the virus is clumping together the red blood cells, okay, which is called hemagglutination. You have an antibody, okay, against that virus. Okay, that would be a neutralization antibody. The antibody can bind to the virus and prevent the virus from agglutinating the red blood cells. Okay, here's some more neutralization reactions. Okay, so here's a toxin that's binding to a cell and causing cellular damage. But if you have a neutralization antibody, that antibody would bind to the toxin molecule before it could bind to the cell and cause its effect. Okay, so this is when we talked about passive artificially acquired immunity. Okay, this would be like, say, for instance, if someone was bit by a rattlesnake and and they needed to get some type of antivenin. Okay, they could get antibodies to the toxins in that venom injected into them. Okay, so it'd be the passive transfer, and it would be artificial. It would be an artificial situation. And those antibodies, that antivenin, would bind to the toxin molecules before it could bind to cells and cause some type of effect or damage those cells. Okay, I think I mentioned to you that those antibodies are actually raised in another animal. Okay, usually I think for rattlesnake antivenin, that is the horse. And then they just take the horse's plasma and purify the antibodies from the plasma, and they have the antivenin that they can use. All right. This is actually looking at the fluorescent antibody technique, or immunofluorescence, okay, which we talked about when we talked about microscopy, right? So this would be using the UV light microscope, and this is immunofluorescence. So there's a couple of different ways to do it. In this particular way, okay, would be referred to as a direct test, because we are looking for the antigen. Okay, so here, okay, if somebody has group A streptococci, okay, like Streptococcus pyogenes, in their throat, okay, and you take the sample, all right, and then you use a fluorescent antibody, okay, that is that this antibody is specifically raised against that particular species. Okay, so it's specific for it, and it's coupled to a fluorescent dye. All right, when that antibody, if that antibody binds to that particular microbe, you have identification, okay, because that antibody is specific. Okay, so whatever species that antibody was raised against, that's going to be the only species that it's going to bind to, and because it's that specific, and so it will give you identification of the species of bacteria that is in your throat. Okay, so this is a direct test, because we are testing for the antigen, but you can also do microscope. Okay, you would see the spirochete. Okay, so you would see fluorescent spirochetes when you look at this using immunofluorescence or the fluorescent antibody technique using the UV light microscope. Okay, so another important immunological test would be the ELISA test, okay, the enzyme-linked immunosorbent assay. And in this test, an enzyme is linked to an antibody, and then that if the antibody is present and which has the enzyme, and you add a substrate which is colorless, the enzyme will turn that colorless substrate into a colored product, and then you know that the antibody is binding to the antigen, and you know that you have a positive test. You can also do the ELISA as a direct ELISA or an indirect ELISA. Okay, so with the direct ELISA, you're detecting antigen, right? That's what you do with a direct test. With the indirect ELISA, you are detecting antibody. Okay, so this would be a direct test, right? Because, okay, we've got antibody bound to a well, okay? And then from the patient, okay, we're trying to detect antigen, okay, the microbe. All right, now for this test, you actually have to have two antibodies, okay? The one would be bound to the well, okay? The other one would be bound to the enzyme, okay? All right, so you add the patient's sample, okay? And just try to imagine if the patient, this is a positive reaction that we're showing here, but if the patient doesn't have the antigen, nothing binds, right? Okay, and then in the next step, you add the enzyme linked antibody, okay, and in this well, number 3, it's binding, right, because the antigen is present. But if the antigen is not present, okay, that second antibody doesn't bind. Okay, you wash the well. Okay, that washes away. And then in the next step, you would add this colorless substrate, and it will turn, if the antibody with the enzyme is present, the enzyme will turn the colorless substrate a color, okay? And you can just look at the well, and if it changes from a clear solution to, usually it's blue, a bright blue, then you know that it's positive. Okay, now if it's negative, that second antibody was washed away, right? So you add the colorless substrate, there's no enzyme to turn the solution a color. Okay, so it would just remain colorless. Okay, I changed the screen just so that you know. This is the indirect test. Okay, it kind of looks the same, but if you look closely, now you've got antigen adsorbed, okay, bound to the well. Okay, and in the next step, you're going to add antibodies from the patient, okay, if they're present, right? Okay, and then so if the patient has the antibody, it binds to the antigen, and then you would use one of those antibodies. Okay, so it's an antibody directed against the constant region of the antibody, and that one would be linked to the enzyme. Okay, so again, if the patient has the antibodies, it would be bound to the antigen, and then the second antibody would bind. Okay, and then you add the colorless substrate, and the enzyme would turn the colorless substrate into a color. Okay, and so this would be a positive indirect ELISA. These pregnancy tests are actually made with monoclonal antibodies. It impresses me that such a sophisticated test can be sold for like $1 at the dollar store. But they have monoclonal antibodies in them, and they actually have two monoclonal antibodies in them. You have to have one directed against one epitope of the HCG, okay, the pregnancy hormone that's found in urine, and then you need to have a second antibody that is directed against a different epitope. Okay, and the idea is that you the urine, you add the urine to one end of this test, and by capillary action, the urine is drawn through the test strip. And on the first, as the urine is going, being drawn by capillary action through the strip, okay, it first hits these antibodies, okay, and they're actually the antibodies that are linked to the color that are directed against the HCG. Okay, so they might be bound to the HCG if the person's pregnant. Okay, if they're not bound to the HCG, okay, they're still going to travel through the strip. Okay, so they're mobile. All right, and then okay, they hit the first. Okay, you see the lines, right? Okay, the bottom line is the test line. The top line is the control. All right. So they hit the first test strip, okay? And there's, on that test strip, you've got the second antibody to the HCG, and it's not mobile, okay? It doesn't move from that line, okay, that position. Now, if the antibody with the HCG is like, kind of floating past with being drawn through the strip by capillary action, and it's going past that other antibody, the other antibody will bind to it and keep it there. Okay, now remember, the mobile antibody is the one that has the color. Okay, so then you're going to see a color. Okay, you're going to see a line. You'll see the red line if the HCG is present. Okay, now there's a lot of that antibody, so not all of it is bound to the HCG. So some of that is going to travel past, okay, the immobile antibody against the HCG, and keep going to the control line. Okay, and at the control line, you've got antibodies against the constant region. Okay, these are the antibody antibodies. Okay, so they would be, so they're going to bind to this antibody no matter what. Okay, and that's the control. Okay, and the control has got to work for you to be able to trust this test. Okay, so it's variable whether or not the test window works. Okay, that's dependent on whether the HCG is present, but the control window, okay, that's just the antibody being bound to by the antibody that would bind to the stem. Okay, so that's the control, and that one always has to light up for the test to be trustworthy. Okay, so pretty sophisticated for a test that you can buy at the dollar store for, $1. Okay, so just to remind you, the direct tests are detecting antigen. Okay, so that pregnancy test that we just looked at, that is a direct test. Okay, The indirect tests would be detecting antibodies. Okay, so we saw how you can do a lot of these tests. They can be either direct or indirect, and I know that the lab exercise that you do asks you about that, right? It asks you whether the tests that you did were direct tests or indirect tests. Okay, and then here are some just some terms, right? Precipitation, okay, like the precipitin ring test, that's for soluble antigens. Agglutination is for particulate antigens. Okay, the examples that we looked at were cellular. Hemagglutination would be the agglutination of red blood cells. Okay, neutralization, okay, via antibodies, okay, would bind to the toxin or the virus and inactivate it. The fluorescent antibody technique, okay, that's immunofluorescence. We didn't talk about complement fixation, but the ELISA test, it uses a peroxidase enzyme as the indicator. Okay, so this was just a short lecture on the immunological diagnostic test.